Welcome to the Chapter 12, Section 1 podcast, and this is on DNA. And let's get started on how it was discovered. First person we're going to talk about is Frederick Griffith, and he was looking at bacteria that cause pneumonia and isolated two strains of the bacteria. And you may have heard this one before. One was the smooth colonies, and the other colony was the rough edges. Basically, the smooth colony had a coating on the outside of it, and the rough edge colony didn't. So he set up 4 individual experiments. In the first one, he injected mice with the disease-causing strain of the bacteria, and the mice died. This is the smooth colonies, and the mice died. Then he injected them with the rough colonies, and these mice didn't get sick. Then he heated up the smooth colonies to kill the smooth colonies, and he injected those in the mice, and the mice survived. Then what he did was he took the heat-killed smooth colonies and then the rough colonies, and neither of these killed the mice in the first place, but when he put them in there together in the mouse died. And not only that, he could get disease-causing bacteria out of the dead mouse. So he concluded that the heat-killed bacteria passed the disease-causing ability to the harmless strain, and he called this process transformation, because the one strain of bacteria had been transformed into the disease-causing one, and he said that there had to be a factor that must contain information that could change the harmless bacteria into disease-causing bacteria. Now, gentleman Oswald Avery, there he is right there, and this is his team. They repeated his work to determine which molecule was most important for this transformation. So they took the heat-killed bacteria and treated it with enzymes. And the enzymes destroyed all kinds of different parts of the cell, including the proteins, lipids, carbohydrates, and nucleic acid RNA. The one thing you'll notice that wasn't included in that is DNA. So what he found was when he destroyed all those things except for the DNA, transformation still occurred. Then what he did was he took an enzyme that broke down DNA, and when he broke down DNA, he found when the DNA was destroyed, transformation did not occur. Therefore, he concluded that DNA was the transforming factor. What did scientists discover about the relationship between genes and DNA? Avery and other scientists discovered that the nucleic acid DNA stores and transmits the genetic information from one generation of an organism to the next. Now, these experiments took place actually at Cold Spring Harbor, and if I remember correctly, Hershey and Chase won a Nobel Prize for this. And they worked on viruses, which are non-living particles that are smaller than a cell, but they infect living organisms. Here's a picture of a virus that infects bacteria known as bacteriophage, and here's the structure of it. And here are some bacteriophages attacking the outside of a bacteria cell. And these viruses are injecting something into the cell. So bacteriophages are composed of two things: DNA or RNA, and a protein coat. The DNA and RNA are found in the head of the bacteriophage, and the rest of the bacteriophage is protein. So when the bacteriophage enters a bacterium, the virus attaches to the cell and injects its genetic information, and then the viral genes produce new bacteriophages, which eventually destroy the bacterium. Here they are bursting out after they've been reproduced. Basically, the bacteriophage hijacks the bacteria to make new virus cells, and then those virus cells go on and infect other cells. So when the cell splits open, hundreds of new viruses burst out. Like I said, this is what's showing here. So what Hershey and Chase wanted to do was figure out what part of the virus entered the cell, either the DNA, protein or the DNA. So they put viruses in 2 different radioactive isotopes, and one stuck to the protein. Sulfur was incorporated into the protein. And when they did that, they found that when they made the bacteriophage with the radioactive sulfur. No radioactivity was found inside the bacterium, so they were sure that the protein coat was not injected. Then they infected the bacteriophage with phosphorus 32 in the DNA, and when the phage infected the bacterium, they found radioactivity inside the bacterium. So therefore, what they concluded was that the genetic material of the bacteriophage was DNA and not protein. So what is the overall structure of the DNA molecule? Well, we got to go into the history of it. Here's a picture of it, which is kind of giving things away, but let's talk about what they needed to do. It had to do three things: carry information one generation to the next. You had to be able to use the genes to determine the characteristics of the organisms, and genes had to be easily copied. Meaning there had to be a very simple, elegant way for this molecule to work. Now, Erwin Chargaff discovered that the percentages of guanine and cytosine bases are almost equal in DNA, and he found that the percentages of adenine and thymine are almost equal in any sample of DNA. And here's some data that was discovered, and you can see there are four different organisms here: yeast, herring, human, streptococcus, and you can see that the percentages of adenine to thymine are pretty close, and guanine to cytosine are pretty close. They're in their neighborhood, and you can see, if you look through all the columns, you can see that. Rosalind Franklin is the tragic figure of discovery of DNA. What she did was used a technique called X-ray diffraction to get information about the structure of DNA, and she aimed these beams at the DNA and looked at the pattern that they recorded on X-ray film. And the photograph up here is known as photograph 51, which was the photograph used to discover that the DNA's true structure. So James Watson and Francis Crick built a model that explained how DNA carried information by using Rosalind Franklin's pattern. They realized it was a double helix, which is two strands that are wound around each other. Here's a picture of their model, and it was shaped like a double helix or a twisted ladder. And James Watson, right here, went on to become the president of Cold Spring Harbor Laboratory, which is not that far from us. DNA is made up of nucleotides, which we've talked about before in the chemistry chapter. It has a monomer of a 5-carbon sugar. For DNA, it's deoxyribose, a phosphate group right here, and then a nitrogenous base. And this is where the information is stored, is in the nitrogen base. And there are 4 kinds of nitrogenous bases in DNA: adenine, guanine, cytosine, and thymine. And you should know all these. So the backbone of the DNA is formed by the sugar and the phosphate groups alternating down the backbone, and they hold the nucleotides. And the nitrogen bases of each of these nucleotides, and the nucleotides can be stuck together. So here you can see there are hydrogen bonds between guanine and cytosine. They form 3 bonds, and then adenine and thymine form 2 bonds. Here's the sugar phosphate bone running down here, and these are hydrogen bonds which are very easy to break, which will become important when we talk about transcription and translation and replication, actually, for that matter. So they discovered that that the base pairs, because the base pairs cytosine and guanine could form base pairs, but they can't form base pairs with the other 2 bases. They were cytosine always bonds to guanine. We call this principle base pairing. So here's a look at Watson and Crick's paper, and you can see it's actually really short. That's incredibly short for probably one of the most important scientific papers in history. And they were awarded a Nobel Prize for the discovery. And Rosalind Franklin died of ovarian cancer in '58, 4 years before they won the Nobel Prize, and you can't give it posthumously. So she was never really truly recognized for what her contributions were. And now let's move on to the quiz.